Hello, ladies and gentlemen. I'm Dan Montano. I'm the Chief Executive Officer of Zhittya Medicine. We're here to give a presentation on heart disease, stroke, Parkinson's disease. Is therapeutic angiogenesis a new medical breakthrough treatment? Dr. Jack Jacobs, my partner, will be giving the medical side of the presentation, but I'm going to make a brief introduction. Zhittya is the Ukrainian word for life. So our company name, Angiogenesis, means life's new beginning. We have been on this journey of discovery, Dr. Jacobs and I, for 22 years. We have expended in excess of $140 million, and we are developing our medicine for 19 medical indications. 19 medical indications that we believe are responsible for the cause of death and suffering for over half of all of the adults on Earth. Now, we want to thank A4M, American Academy of Anti-Aging Physicians, for giving us the opportunity to make you aware of what we're doing. So let me clarify something. We have no product for sale. We have no service to sell you. Our drugs have not been approved by the FDA. We are going through the clinical trial process now to get approval. Dr. Jacob will show you some of the amazing data that we've achieved. And we have no services. We do not take donations. So all we're here to do is to make you aware of our medicine, make you aware of what we think we can do. And perhaps in a clinical trial, you may know a patient you can refer to our clinical trials, or maybe you may think of some ideas that we haven't thought of or some ways we can advance. After we obtain US FDA approval, then we'll be looking to physicians like you to help us disperse and sell the product and to provide the services to clinics all over the United States, to practices all over the world. We believe that these drugs that we are developing in 16 of the 19 indications are unmet medical needs. Parkinson's disease has no treatment to cure it. It has no treatment that is really very good. We believe we can address the root cause and reverse it. We believe that about 16 out of the 19 indications Dr. Jacobs is going to present to you. So what I'm going to do now is present my partner, Dr. Jack Jacobs. Jack has his PhD from Washington University in St. Louis, WashU as they like to call it. Jack was one of the people that originally helped identify this molecule almost 50 years ago when he was a graduate student, which is extremely impressive because he keeps telling me he's 43. Talk about anti-aging. And Jack went to work at Hitachi Pharmaceutical, where he set up their biologic operations, and then he went to Merck, where this molecule was originally licensed, and he worked at Merck and their biological activities. Jack and I, as I said, started together for 22 years. He is the science partner, and I'm the cute one. Ladies and gentlemen, Dr. Jack Jacobs.
All right. Thank you, Dan, as always, for that very nice introduction. So I'm going to be talking about this process of angiogenesis. Angio is blood vessels, and genesis is new blood vessels. So we have a drug which stimulates new blood vessels in tissues where the blood circulation needs a boost, such as in heart disease, stroke, and I'll talk about Parkinson's disease. Now we have over 60,000 miles of blood vessels in our bodies. These are all formed by this process of angiogenesis. And if we get blockages in our vessels, such as in the heart, I'll talk about stroke, we have blockages in the vessels leading to our brain. And then recent data indicates a number of neurodegenerative diseases, such as Parkinson's disease, Alzheimer's, all are initiated by a lack of blood flow in a certain area of the brain. So let's talk a bit more about angiogenesis. And actually, over 75 human diseases result from a lack of angiogenesis or a lack of blood flow to a tissue or an organ. And Dan mentioned that we're working on 19 of these. I won't talk about all 19, but at the end, I'll talk about where you can get more information on these 19 different indications. But again, the easiest way to get more blood into a starving tissue, let's say a starving heart tissue, people have heart disease, their coronary arteries are blocked, is to grow new blood vessels through this process of angiogenesis. And that's what our drug does. It's the most potent stimulator of new blood vessel growth in our bodies. This is a natural process, and we use it every day to heal, for example, cuts and scrapes. This skateboarder has fallen off his skateboard, scraped himself, and he's going to use our drug and this process of angiogenesis to heal that wound. If we look at this a little bit closer, say this is his elbow, this is the scrape, the scab, and this white circle here. We look underneath that, and here we can see angiogenesis at work. These are all brand new blood vessels. And what they do is bring lots of different factors which can regenerate this wounded area. And it regenerates not only skin, but anything that's needed, hair follicles, sweat glands, new nerves. And the molecule in our bodies that's stimulating this new blood vessel growth is our protein, I'll talk about in a second, FGF1. Here it is. It's a protein, so a protein like insulin in our bodies, like human growth hormone in our bodies. So this is another growth factor. Dan mentioned I was involved in purifying this not 50 years ago, a number of years ago in Washington University. We got it back then from cow brains. We would go to the St. Louis slaughterhouses and get cow brains, but now we use in our clinical trials a human recombinant form. That's the way most proteins are made for clinical trials and then commercial development. Now, as I said, this is one of the most potent stimulators of new blood vessel growth in our bodies. And let's take a look at this. So let's say this skateboarder fell off his skateboard, cut his elbow. Immediately, his body senses that he's wounded and begins making FGF1. And that's shown here, these little guys flying in. And so these new vessels form off of existing vessels. So the FGF1 comes and lands on its receptor. This is the FGF1 receptor. This is a very important interaction. It's like a key going into a lock. And that's going to stimulate the production of a very important cell, the endothelial cells. These are the cells that make up our blood vessels. And they start dividing. This is all due to the action of our drug, FGF1. Here they're dividing. They actually start chewing a little hole in this capillary, and then they start forming a tube. And here they're elongating, sending blood vessels into the wounded area in his elbow. And that's going to provide more blood perfusion to get that wound healed. Now, an important concept is that only when you have injury do you get an upregulation of these receptors. So if you put FGF1 into normal tissue, nothing's going to happen. But we know in heart disease, in stroke, we get a tremendous upregulation of these receptors, and that's needed to stimulate this new blood vessel growth. So if you put our drug into normal tissue, healthy tissue, nothing's going to happen. Okay, again, we're developing this for 19 different medical indications. I'm going to talk about 3 today. I'll start off with coronary artery disease, and then we'll move to the brain, talk about stroke, and end up with Parkinson's disease. And at the very end, we're going to talk a little bit about aging and how we can use our molecule, we think, to reverse the deterioration of our vasculature as we age. Okay, coronary artery disease, we'll talk about injecting the growth factor directly into the heart, but just let me give you a few statistics on heart disease. It is the leading cause of death in the United States. 23.4% of people will die from heart disease. This actually is a little bit down from 10 years ago, probably due to better diet, more exercising, but still the leading cause of death in the United States. Surprisingly, if you ask people what's the leading cause of death in women in the United States, heart disease kills more women than all forms of cancer, which to me was very surprising. So again, the leading cause of death of both men and women in the United States. Now let's look at the aging heart. This is, let's say, a 70-year-old who is healthy, but as we age, we all develop calcium deposits in our coronary arteries. This isn't the heart, this is the coronary artery. And we get blockages, as shown here. This is a blocked coronary artery. And if these new vessels didn't form, that's called collateral circulation. So our body naturally, if a vessel gets blocked, sends out signals to develop more circulation, more blood vessels to supply the area of the heart. In people with heart disease, this process does not happen. So they get a blockage, and for whatever reason, they don't have enough growth factors to stimulate this collateral vessel formation. And what happens is this muscle, without perfusion, starts to weaken, and it weakens, and it can lead to angina, which is chest pain, or it can lead to a heart attack. So what we're going to do in our clinical trials, we actually injected FGF1 into people that had bad heart disease, and I'm going to show you a video where you can see these patients actually grew brand new blood vessels 3 months after we treated them. We've done 3 separate studies, 2 in Germany, which I won't talk about, but the one in the US I know a lot about. I supervised this trial, and we got some very nice results. Patients did very well in this US trial, so let me talk a bit more about that. We had 6 sites, and one of the sites, the University of Cincinnati site, did really well. They recruited most of our patients. The patients did very well and attracted media attention. So ABC News went down to our Cincinnati site, interviewed our cardiologist. You'll see her, Dr. Lynn Wagner, and some of the patients who got our treatment down there. And I'll show you the news clip. It's a short video, but please pay attention to when the doctor, Dr. Lynn Wagner, points to the new blood vessels you can see growing in these patients' heart after they were treated with our drug.
But since receiving an experimental treatment for his blocked arteries, his pain is gone.
I really feel great.
Duke was one of the first heart patients in the country to be treated with a protein actually capable of growing brand new arteries. The genetically engineered protein is injected directly into the heart. Within days, a network of new vessels begins to grow around the blockage, increasing the blood supply. Dr. Lynn Wagner showed us the changes in one patient's heart.
We see a small, narrow main artery and not very many secondary and tertiary arteries. This is after the treatment. What we're now seeing is new blood vessels growing here off the end of this artery.
And the patients themselves?
Symptomatically, they're improved within a couple of weeks of the treatment.
Just ask Constance Donnelly.
Oh, I feel wonderful. I've never felt so good in the last 5 years.
But doctors already see potential in other cases where the blood supply needs a boost, such as strokes and diabetes.
Okay, let's now move on to stroke recovery. And I'm going to show you some nice data in animals, and we're submitting an application to start our human studies, hopefully in about 4 to 6 months. So our brain is incredibly vascularized. We have these large vessels, we have lots of small vessels, billions of blood vessels in our brain supplying billions of these neurons. And obviously these neurons are very active. They need lots of oxygen, lots of glucose, and they also generate, as all cells do, metabolic waste products. So the vasculature actually, gets rid of those waste products, and sometimes they build up, and we'll talk about that in Parkinson's, where you get accumulation of toxic proteins in that disease, much as you do in Alzheimer's disease, where you see the plaques. Now, we know if you get a blockage in one of these main arteries, you can get an acute stroke, and sometimes people also get very many mini strokes in these smaller vessels, and as they accumulate over time, this can lead to problems as well. But let's look at this acute stroke. A clot develops. This is the carotid artery leading into the brain of a person. That causes a stroke, causes damage to a particular area of the brain. And depending on where the area is, people have different disabilities after a stroke. This shows a brain slice. I'm going to show you some of these in animals, but this is a slice of the brain, and here is the stroke volume, it's called. This is comprised of dead neurons, and these neurons in pink are at risk of dying unless they get better blood perfusion. And that's what our drug is going to do. It's going to come in here, grow new blood vessels, and actually regenerate that tissue. Let me show you some slices from an animal experiment. This animal was given a stroke. This is a mice. here is the stroke area. So we tied off an artery in the mouse's brain. They developed a stroke. Second group of animals, same thing, but we gave them FGF1, our drug, intravenously for a 2-week period. And you can see, I think, the remarkable difference between no drug and animals treated with FGF1. Now, we can look into the brains of these animals and see what's going on. Are we growing new blood vessels? Are we making new neurons? Do the mice do better on behavioral tests? So let's first look at blood vessel. So here we're looking at capillaries in the mouse brain. Okay, we're not looking at neurons now, but there are millions of neurons in this field of view. But here are all these fine capillaries which are supplying the neurons with nutrient. We tie off an artery in the brain, and you can see 24 hours later this decimation of the vasculature. So this is the stroke area. We treat one group of animals with FGF1, another group gets placebo, and you can see here after 2 weeks this what I think is a remarkable and significant regrowth of these capillaries. Not quite back to normal, but look at it compared to the placebo treated. This brain is desperately trying to get blood into this area, trying to grow new blood vessels. This is disordered growth. There's a tangle here, and this actually can hemorrhage, and the mice actually do worse. Now, to look at the growth of new neurons, we look at their motor activity. So if these mice have damage, they're not going to do well on motor skill tests, and we put them on it's a rotating rod. So this rod is rotating, and here normal mice can hang on, as you see here. The animals that had a stroke and got FGF1, they're having, they're hanging on pretty well here. They're, not quite here, but they're certainly better than the placebo guys who have fallen off the rod and we can't even see them. So from these animal experiments, we see we're growing new blood vessels with FGF1 treatment. We're growing new neurons, which lead to this motor skill improvement. And we're very excited about taking this into people who have had a stroke, who have stabilized, and we're going to see if we can get back function in our first clinical trial that we'll be doing. Okay, let me talk about Parkinson's disease now. Work from a number of medical centers have recently shown, they're using perfusion, blood perfusion testing, that Parkinson's disease is really a vascular disease, that it initiates by a lack of blood flow to a very small area of the brain, which is affected in Parkinson's disease. So what happens in Parkinson's is dopamine neurons. This is a very specialized type of neuron, which is involved in movement, motor skills in people. We're only born with about 500,000 of these neurons, as shown here. And healthy aging, we think we have a lack of blood flow, which leads to a decline of these dopamine cells in healthy individuals, but they never really lose the amount that people with Parkinson's disease do, as shown in blue here. So here we see the inexorable decline of these dopamine neurons in patients with Parkinson's. When they lose about 60 to 80% of their dopamine neurons, they start displaying all the classical symptoms of Parkinson's, the tremors, the gait disturbances. So in our clinical trial, we hope to be able to regenerate enough of these dopamine neurons to bring them back up into this region and hopefully reverse their symptoms. And I'm going to show you some very nice data in monkeys where we did that, and that formed the basis for our regulatory submissions to do this same thing in people. In Parkinson's disease, these dopamine neurons are in a very tiny area deep within our midbrain. It's called the substantia nigra because nigra is black and these dopamine cells are very dark, as shown here. They become dysfunctional in Parkinson's, we believe due to a lack of blood perfusion in this area, and I'll show you some data on that in a second. Leads to the normal dopamine neurons becoming dysfunctional, secreting less dopamine, leading to the movement disorder seen in Parkinson's. So let's look at some blood perfusion data in Parkinson's disease. This is on patients now. And what they did is they looked with a what's called functional MRI. And this is in real time, you can see blood perfusion in different areas of the brain. So they focused the MRI on that substantia nigra region where the dopamine cells are. And you look at healthy 21-year-olds, you have maximal blood flow in that very small area of the brain. I showed you as we get older, we lose some of our dopamine neurons, not enough to develop the symptoms of Parkinson's, but they're down about 25% in a healthy 65-year-old. This, again, is blood perfusion. But look at Parkinson's disease here. This functional MRI indicated about a 50% reduction in blood flow in the area which is perfusing all those dopamine neurons. So they're seeing a lot less blood flow, we believe, a lot less nutrients. We believe this is why they become dysfunctional. And if we can increase blood flow to that region, we believe we can bring these people back. Let me show you another blood perfusion study. This is a woman who had both Parkinson's and dementia. And here we're looking at the areas of the brain involved in dementia: memory, executive functioning, and cognition. So this is the entire kind of top of the brain. It's called the cortex region, both halves. And in blue here, we see normal blood perfusion. Yellow is reduced, and this red area, severely reduced blood perfusion in these areas of the woman's brain that control memory, cognition. So we believe if we treated a woman like this with FGF1, we would not only hopefully increase perfusion where her dopamine neurons are, but also in these memory type neurons, and that we could also improve her dementia. We have some nice data in a monkey model of Parkinson's disease. This is more or less the gold standard to test drugs before they go into the clinic. You can give a monkey Parkinson's by injecting a toxin. It's actually a herbicide directly into the brain. And over a period of about 6 months, he'll come down with all the classical symptoms of Parkinson's. So here's the results of such a test. So this is motor score. So 4 is normal. 1 is highly abnormal motor skills. And here's this toxin that's injected into the brains of the animals. This is a herbicide. And just as an aside, a very tragic story. On the 80s, a marijuana farmer sprayed his crop with this same toxin. At night, some teenagers broke in, smoked the marijuana, and came down with irreversible Parkinson's. So a real tragedy, but the same toxin is the one that is now used in this animal model. And after about 9 months, right here, these animals are displaying all the motor symptoms, the classical motor symptoms of Parkinson's. So they're tremoring, they don't move very well. So we start at this point treating one group in blue with FGF1. This group in red gets a placebo dose. And you can see we're treating them over about a 6 to 7 months period. And we get out to the end of here, you can see the movement scores of these treated animals with our drug are about, I'd say 80% back to normal. Maybe if we'd gone a bit longer, we could have gotten back to normal. But clearly a significant difference between the placebo-treated animals that are really are struggling with their movement. Now we can look in the brains of these animals and ask questions. What's happening with the dopamine neurons? Are we attacking the root cause of Parkinson's, which are these dopamine neurons becoming dysfunctional? And indeed, when we look in the brains of these animals, in the substantia nigra region where the dopamine cells are located, the new regenerated dopamine neurons stain brown. So you can here see a very nice, rich brown staining here. These are all brand new dopamine neurons. In the placebo-treated animals, you don't see that at all. So we believe these FGF1 regenerating dopamine neurons kind of reversing the root cause of this disease, and these animals are moving much better. Now, one other thing we was looked at is the accumulation I mentioned of toxic proteins in this region of the brain. Similar to Alzheimer's, we get beta amyloid plaques in people, in patients, and monkeys with Parkinson's. You get what's called aggregated synuclein. So this is a normal protein which for some reason begins aggregating in this disease. And here you can see it here. This is an aggregated synuclein protein which is toxic to the dopamine neurons in that region of the brain. We believe it accumulates because there's not enough blood perfusion to get it out of that area of the brain. And you can see here in the animals which we treated with FGF1, there's a marked reduction in this toxic aggregated protein. So not quite back to normal, but surely better than this situation. So we think these two very important physiological processes. One, regrowing the dopamine neurons. Two, getting rid of this toxic protein lead to the motor score improvements in those monkeys. And we hope to see the same thing when we start our human trial. So we've designed a clinical trial for people. We're going to look at 3 ascending doses of the FGF1. No placebo groups, so all the patients will receive a 1-hour intravenous infusion of FGF1 5 days a week for 4 weeks. So this is a commitment to the patients entering this trial. We're going to put a central PICC line in so we don't have to be poking the patients every day when we give the drug or when we withdraw blood for lab testing. This is a proof of concept study. This is the first time that FGF1 will be tested in the brains with this Parkinson's disease. We'll be looking at safety. Is our drug safe in the brain? And hopefully we'll start seeing some effectiveness. If we can regrow those dopamine neurons as we did in the monkeys, that we will see improvement in motor skills in the patients. But if not, as long as our drug is safe, we can tweak the trial. We can give more of our FGF1. We can give it for longer periods of time. And we'll certainly follow those patients to make sure the drug is safe in the long term. So where are we with our clinical trials? In the US, we've submitted an application for Parkinson's, also for diabetic foot ulcers, which is another indication. I didn't talk about that, but we are also doing trials in that indication. The FDA has asked us to do 1 additional animal trial, which we're doing now before we can start. Since we were held up a bit with this, we thought, let's try some different countries. And someone came to us and said, Mexico probably will accept your animal data that you have now. So we submitted 2 applications, Parkinson's disease and ALS, which is Lou Gehrig's disease, another neurodegenerative disease, which is thought to be initiated by a lack of blood flow. The Mexican FDA said, fine, your data looks fine, you can start. So we're getting ready to start that. We were held up, unfortunately, by the pandemic, but hopefully we can start that as soon as those restrictions in Mexico have eased. Because of our success in getting trials approved there, we're also going to be submitting in the near future an application to treat patients with Alzheimer's disease, MS, and stroke recovery. We also have an application in Europe, in Estonia, which is part of the EU. They're in their final review now. We're actually getting comments back from the Estonia regulatory people just this week, and we think we'll be able to answer those and get that trial started. And also, we're working with neurologists in Morocco to get a Parkinson's disease trial going there. Now, people ask me, Jack, where are you doing this clinical trials in Mexico? Are you in some tiny clinic in Tijuana? And I say, no, we're in probably one of the best hospitals in all of Latin America. It's called Hospital Zambrano. It's in Monterrey, Mexico, a very modern city. Here's the hospital, new, clean, superbly equipped, and really well staffed. We went down there, Dan and I, to visit and had a real good visit there. We've hired an international clinical research organization, a CRO, Cohortius. They're based in the Netherlands, but they have an office in Monterrey, familiar with the hospital there. So they'll be running the trials down there to US FDA standards. That's a very important point, that we can use the data generated there to help support our trials in the United States. And also patients who want to perhaps come down to Mexico to be treated, they'll know that they're getting a trial run to the highest standards. Our principal investigator is a movement disorder neurologist, Dr. Martinez, trained at the National Institutes of Health in Bethesda, well trained. As Dan mentioned, we pay all the costs for the clinical trial, including travel and housing expenses for our subjects who volunteer, or our warriors, we call them, who volunteer for these trials. And so we don't want donations. We have the money raised to do that. As I mentioned, we'll start when the pandemic restrictions have eased. And as I also mentioned, ALS, or Lou Gehrig's disease, has been approved to start down there as well. Okay, we're developing our drug for 19 different medical indications, all due, we believe, and others, to a lack of blood flow to a tissue or an organ. All that can be treated by a drug which stimulates new blood vessel formation, or angiogenesis. I talked about heart disease. We talked about stroke recovery. We have good animal data, PAD, peripheral artery disease, blockage of arteries in our legs, leading to pain on walking, and in some cases, lower leg amputations. We have a whole host of wound healing disorders, which I didn't talk about. These are very kind of straightforward clinical trials because it's a topical application. We're just applying the drug outside the body on the skin. And what we have FDA clinical trial data, phase 2, where we can heal diabetic foot ulcers and also venous ulcers. So we'll be starting those up as well. And also bed sores, surgical wounds, burns, and kind of serendipity we found if we injected FGF1 under the skin, little tufts of hair grow up. So we're probably going to be looking at male pattern baldness as well. So these are all what we call wound healing disorders. And then we have all these brain disorders that we and others feel start from a lack of blood flow to a specific area of the brain. Parkinson's disease, ALS, Alzheimer's disease, chronic depression. FGF1 levels are severely depressed in people with chronic depression. MS, Victor Montano will be giving a seminar on Monday on this. Traumatic brain injury, post-traumatic stress disorder. CTE is just from repeated concussions or head injury. Boxers, NFL football players come down with that. Vascular dementia and MSA. So all these indications we believe we can treat by growing new blood vessels in the brain or in other tissues. Let me just switch to another way where the vasculature can become dysfunctional. And that's through newer research that the vessels can become leaky, okay? So in some cases, we're growing new blood vessels. In this case, we're going to talk about healing dysfunctional vessels that develop leaks. And again, here are these endothelial cells. These are very important cells that line all of our blood vessels and keep things that should remain in the bloodstream. And we know, for example, in certain diseases such as MS, immune cells get out of the vasculature and actually attack nerve cells that are adjacent. So this is more or less an autoimmune disease. But how do the immune cells get to the nerves? They get there by leaking out of the vasculature. And I'll show you some data where we have evidence in animals that we can heal these leaking vessels. So when these vessels become dysfunctional, and I'll talk about this also, we believe happens in the aging brain, but in certain diseases such as MS, they develop what's called endothelial cell dysfunction. So here's normal endothelial cells. Again, these are the cells lining the inside of our blood vessels. So we're looking inside a blood vessel here. These are tightly packed, well-ordered cells in healthy blood vessels, healthy capillaries. Here's what happens in MS due to genetic and possibly environmental factors. The endothelial cells become dysfunctional. You can see they're bumpy, they're disordered, and they become leaky. And this is where we see leakage out of the vessels of toxic constituents that are in the bloodstream, and also, very importantly, immune cells, which then go ahead and attack neurons and form the lesions that are seen in MS. Now, we looked in a model of MS, an animal model of MS, looking at our FGF1, to see if we could heal the capillaries, heal the endothelial cells damaged in that MS model, animal model. So here you see the inside of a capillary in one of these animals that has experimental MS, the lesions, the leakiness, and we treat here with FGF1. And you can see here day 10, day 15, we're starting to heal. We're starting to heal that endothelial cell layer inside the blood vessel. So where we get out at day 30 here, we've completely healed this blood vessel in this animal model of MS. And these vessels are no longer leaky. We're really excited about that, and we'll be, again, starting a clinical trial in MS, hopefully in 4 to 6 months. Now, I just want to end with some work done on aging, which we think, again, a lack of blood flow, either through blockage of vessels or through leakage through this vascular endothelial cell dysfunction, leads to normal aging of our brains. And why, as we get older, we forget things. We have cognitive decline. And so, very prestigious medical school, Columbia University Medical School in New York City. This was something published in Cell, again, a very peer-reviewed, very prestigious medical journal. And then Time Health did a little video on this and why our brains perform worse as we age. Let me give you the punchline, and I'll show you the short video. What the video is going to show you is that we all have these neural stem cells in our brain, okay, whether we're 90 years old or whether we're 15 years old. But as we age, blood perfusion in our brain decreases, and these neural stem cells become less and less active. And so they're less likely to form new neurons in the memory part of our brains, which this video will show, but even in diseases such as Parkinson's, these dopamine neurons are not going to form as readily if these stem cells are not as active due to a lack of blood flow. Okay, so let me run the video and I'll be back at the end. So we agree completely with this video. We believe we have a drug which can improve blood flow to the brain, whether by growing new blood vessels, as we saw in the stroke models, or healing leaky blood vessels, as we saw in the model of MS. So we're moving ahead. We're going to be testing this molecule in brain disorders we think result from a lack of blood flow, also in the heart, and all those wound healing disorders. At this point, I want to point out we have a website, ZGM Care. You can go there. I've written white papers on most of those 19 medical indications. Importantly, we have webinars that we gave in the past on Parkinson's, on ALS, and you can view those videos at the website. And you can also sign up for our clinical trials if you want to be considered for the clinical trial. You'll get on our mailing list, and we'll keep you updated as to when and where these trials will be started. So at this point, I'd like to bring back Dan, and we're going to take some questions here. It could be science questions, or if you have any business questions, we can answer those as well.
Thank you, Jack. We got a bunch of questions, but first of all, I would just like to emphasize to everybody what Jack said. This is a small booklet we have, which we can make available if you email us. It basically goes through what the medicine is, why it works, and some clinical data. This is an example of one of Jack's white papers. This one's on Parkinson's disease, but we have these, as Jack said, on numerous ones. And also we have a list of, as Jack said, other webinars that we've done that are on YouTube on our Zhittya channel. So we've got a couple of questions here, Jack. Let me start with the one that we always get. Can this growth factor cause or trigger cancer?
Yeah, that's a good question. We get that a lot, and people are, savvy to ask about a growth factor in cancer. But let me tell you this, we've looked very hard at that question, and the FDA made us look very hard. In our animal studies, before we go into people, we tested FGF1 at doses of about 100-fold higher than we're giving to people, and we saw no hint whatsoever of cancer initiation. In our clinical trials, we look very carefully at any tumor markers. We follow, I think, about 5 to 6 of those standard tumor markers. In the over 90 people we've treated, we've never seen a bump up of those tumor markers. So we feel very, very confident that this drug of ours does not at all initiate cancer.
Okay. Jack, this is a question from one of the attendants. Would giving this in a hyperbaric oxygen environment work better?
That's a good question. I think that's something we might try with the diabetic foot ulcers, where there's some indication that oxygen treatment can help heal those wounds. So perhaps FGF1 plus this hyperbaric treatment might might speed up wound healing in that situation.
Okay, we have another question from attendee. Will severe long-term anemia also cause similar effects to the neurons in the brain?
Yeah, long-term anemia could result in lack of oxygen to neurons. And so obviously, yeah, that's a different disorder. That's not due to blood vessels, but due to a lack of iron or red blood cells. And so our drug would not treat that, but anemia could be a cause of neural or neuronal damage.
Okay, next question. What are the side effects of this drug, Jack?
So we've looked carefully. We can say of the 90 people who've gotten this drug, we have not seen a severe adverse effect that we can attribute to the drug. In animals, again, the FDA makes us go to very high levels, and only at the very, very highest doses, maybe 100-fold higher than we're using in our clinical studies. This is a growth factor and things start to grow, and the thyroid gland is sensitive. It starts to grow a bit, but when we stop giving the drug, it reverts back to normal. We've never seen any thyroid changes on any of our patients in our clinical trials.
Okay, hold on a second. Did FGF1 administration bring any benefits on lipid and glucose metabolism in clinic?
We follow cholesterol levels and we look at those labs, and no, it did not lower cholesterol in the patients that we treated. And in fact, we found that with very high cholesterol levels, that seems to inhibit a bit the activity of our drug. So we really ask people to get their cholesterol under control before they enter our clinical trials.
Okay, next question. Why don't blood vessels start growing all over the body when FGF1 is given?
Good question. And I kind of alluded to that, but again, when the body gets damaged, it upregulates the receptor. That's the kind of docking mechanism, about 100-fold. And only when that happens do you get new blood vessel growth. So that'll happen in the heart when it becomes starved or ischemic, and we believe in the brain areas affected by Parkinson's or ALS. So if you inject again this drug in the normal tissue, nothing happens.
Next question. The PICC line, would that be all the way into the brain?
The PICC line goes up through the elbow and goes close to the heart. So it ends up in the vena cava, which is one of the major veins, gets into the heart and then immediately gets pumped up towards the brain. So it's a way of getting, we think, higher drug concentrations closer to the brain, not as diluted. And also, it's to be stuck every day with a needle is not a lot of fun.
Okay, Jack, there's a question here that's a rather complex one, so I'm going to try and rephrase it. The question is there a time period when a person has had the disease where there is no improvement with your treatment, a point of no return?
Well, we don't know that yet. We're just starting these trials. In all the trials, we're treating mild to moderately severe disease. So obviously, people with very severe Parkinson's or Alzheimer's really would have trouble making it to the clinic to be treated. But we're going to be looking at a diverse population, and hopefully it'll work at most stages, but we just don't know the answer to that yet.
Okay. The next question is leaking in the gut. Would this possibly help with the endothelial cells leaking?
Yeah, we've seen some gastroenterologists talk about problems with ischemia in the gut. So the vasculature gets blocked off, maybe due to blockages, maybe due to leakages. But yeah, I think that's a very additional. We have 19, how many we can do, but that is certainly something worth looking at some point.
Jack, a question here. How long do you believe the drug can rejuvenate the endothelial cells or maintain the outcome that we've attained?
Yeah, so we think the blood vessels will either heal or form new ones. We have most data in the heart. So we looked 3 years after we treated the patients, and the blood vessels we grew 3 years previously were still there, still perfusing the heart. And those patients that had 1 to 2 years to live, some of those were still alive after 8 to 10 years. So we think it's a durable response. But these are neurodegenerative diseases, heart disease, are chronic diseases, and they might need additional treatments in other areas of their heart, or they might, like in MS, might develop lesions outside of where we treated.
Okay, Jack, I'm going to sort of help answer a question that you've already answered, because as Jack will tell you, he is a scientist and he has to prove things. And he's always complaining that, Dan, you can speculate, but you don't have to prove it.
All right.
People we dosed in the heart were end-stage heart patients. They were in the worst condition you could be. The people in the United States basically had to be given by their doctors less than 1 year to live. So we did the very end stage. In the case of diabetic foot ulcers, when you have a chronic ulcer, and Jack didn't show you the data on that, but in these chronic ulcers, and if you've ever seen an open foot ulcer, you know these things can be ugly. We were able to close 100% of these in a period of 5 months, compared to the placebo group with the finest treatment in the world could only close 70%. And if you realize how many millions of people have diabetic foot ulcers, that 30% that doesn't close is a staggering number. And so in the brain, the one thing that I want to make a point, Jack is talking about the angiogenesis factor, getting new blood flows, but we know that stimulates the neural stem cells, the neurogenesis. And I have to believe maybe it's one treatment, maybe it's 3 treatments, maybe we have to come and keep, but I don't believe there will be people that are so lost that we cannot recover the neurons. Now, I want to make sure we understand something. If somebody has a stroke and they're materially disabled, I don't think if we treat them and we repopulate all those neurons, they're not going to get up and play basketball the next day. They're still going to have to be rehabilitated. So a lot of you doctors who work in the area of rehabilitation, that to us is going to be a huge new exploding business, for the simple reason it's one thing to try and help people who are on a long-term decline. It's another thing if we're able to repopulate the neurons, if we're able to reconnect things and give adequate perfusion so the neurons can perform, and then you're teaching people to come back to the life they may have had before, get closer to it. So Jack has to tell you the facts that he can prove, and I can tell you what we're hoping in our hearts. So Jack, here's a question. Would basal cell cancer disqualify somebody from a clinical trial?
No, it would not. That's actually one of the only, you can't have active basal cell, but if it was in your past, that does not disqualify you from being in the trial. That was not my call. That was the call of our principal investigators or physicians talking with cancer people.
Jack, we have another question here. Will this possibly eliminate the pyron proteins in the brain of Alzheimer's patients?
Okay, so it's, yeah, it's not the prion, it's a beta amyloid protein, which the prion is a different protein. Yeah, we think, as we saw in the Parkinson's monkeys, we got rid of that aggregate, and it's very similar to the beta amyloid plaque, which accumulates in Alzheimer's disease. And we think if we reestablish blood flow, we can start flushing that stuff out. That's our hope.
Now, Jack, we have one more question here. And that question is from somebody regarding the repopulation and the stroke enhancement. When do you know the criteria for people to be in that trial?
Yeah, so for that first trial, which we hope to get going in 4 to 6 months, so it's not someone who's having a stroke. We want someone who's had a stroke, and then we know that it takes about 6 months for those patients to stabilize. They might get better, but they usually reach a plateau at about 6 months. And that's where we want to start treating them. So I'd say 6 months or greater. I'm not sure what the outer time limit will be, but not before 6 months. So it's really truly a stroke recovery type of clinical trial, not treating the initial stroke, let's say 4 hours after someone comes in with a stroke.
Okay, Jack, we're down to our last 4 minutes to stay within our time limits. So I would like to sort of make a closing statement. I'm going to do it in 2 parts. One of the questions that we get from people is, why are we doing a clinical trial in Mexico? Are we trying to hide from the regulators? Why are you considering Estonia? Why are you looking at doing a Parkinson trial in Morocco? The US FDA has requested that we do additional animal data to put our molecule in the brain. The data that we had on animals for the brain, the FDA said they wanted something more refined, more sophisticated. As you know, new things are discovered every week, and the FDA is constantly wanting the 10,000th trillionth now compared to what they accepted 5 years ago. In Mexico, they accepted our animal data, which means we can be dosing people. And the question is, can we put the molecule into people, infuse it into people, and have it clear the blood-brain barrier? And can that be safe at level 1, level 2, level 3? Once we know it's safe, then we have a lot more latitude on things we can do. If we dose people in Parkinson's at level 3, which is the level Jack believes will be something that equates to what the monkeys got, and we see outcome, that is wonderful. Now, if we do the clinical trial in Mexico and find out the molecule is not safe in the brain or has no outcome, why do we need to spend $5 million in the United States to prove it doesn't work? If we have evidence in Mexico it doesn't work, then it doesn't work. But we think these neurodegenerative diseases are so critical, and time is against these people that are suffering with this, whether it's Alzheimer's or stroke or Parkinson's or multiple sclerosis, that we think time is of the essence. And we think that in the case of Parkinson's, if we can be dosing in the first quarter of next year, we can know by this time next year if it's safe and if it works. That to us is important news. We have the group in Estonia that came to us. They said they would accept the animal data. Let's try it. It's a former Soviet country that got its independence. The drugs are approved at the Central Tallinn Hospital or East Central Tallinn Hospital. Jack and I were in Estonia, by the way. Beautiful country. We were approached by a group of neurologists. Jack's giving a webinar presentation to 3,000 neurologists in the Middle East and Africa for the Moroccan Neurologist Conference next month. What we're doing is cutting edge, but they wanted to do the Parkinson's. They said that if their hospital has the authority from their government to approve drugs, Jack has submitted the application. We're waiting for their response. Are they going to start the clinic or clear the clinical trial? But when I asked them the question, when did your hospital get the permission from your government to approve medicines in Morocco? And the response was he wasn't positive, but he thinks it was 840 AD or 850 AD. Some of us forget there was a world before we came along. The reason I say that is we're doing these not because we're trying to avoid everything. Everything's being done to US FDA standard. We're doing this because we want answers fast for people that are suffering. I got a call from a doctor who's on the A4M that we met last year, 75 years old. He said, Dan, I've got Parkinson's. How can I help? So besides you treating other people, a lot of you want us to succeed also. So we want to thank you for your time, ladies and gentlemen. I think, Jack, hold on. We still got 42 seconds, Jack. Anything you want to say?
No, thank you for attending, and follow us at our website.
Make sure you follow us, even if you don't like us. What we're doing could be cutting edge and could add 20, 30 years to your life. Have a good night, and may God bless all of you. Bye-bye.
Goodbye.